Hey everybody, I hope you're doing well at home. I certainly miss seeing you here in the classroom. In lieu of not being in the classroom today, I'm going to go over some principles of electrophoresis. Remember last week you guys, in a hurry, you ran a PCR, a polymerase chain reaction, which takes a segment of DNA and amplifies it, makes lots of copies. And remember what's inside of those little tubes is just a little bit of liquid. So when in biotech we run into a problem and we want to know, well, hey, did our PCR actually work? We want to confirm whether or not we actually amplified the correct DNA, if we amplified any DNA, and we're not going to be able to see that just by looking at the tube. So this next technique allows us to confirm whether or not we actually have a targeted sequence of DNA. Like, did we actually get it? Right? So this is the step in between PCR and sending your DNA off for sequencing. Today, what I just want to focus on is how does this technology work? And then what we'll do later in the week is we'll look at what are your results? You know, what did you get, essentially? So there's some concepts that we have to review here first, and that's the basis for DNA electrophoresis, for gel electrophoresis. And the big key here is that we're going to take advantage of DNA being negatively charged. And your question might be, well, I don't remember, why is DNA negatively charged? Well, we've got three pieces here of a nucleotide of DNA. What we've got is an adenine nitrogenous base. We've got this pentose sugar, the 1', 2', 3', 4', and 5' sugar, and attached to that 5' carbon in the sugar, we've got this group called a phosphate group, remember, which is part of the backbone of DNA along with the sugar. And these two are bonded together, right? Well, in an aqueous environment or in a water-filled environment like DNA is in all of the time, whether that's in cells or it's in a tube, this phosphate group is negatively charged. Right? So you see these little oxygen atoms here have a negative charge on them, right? And overall, that's going to give DNA a negative charge. If there's lots of phosphate groups like there are on a strand of DNA, it will actually have a pretty significant negative charge to it. So that's the thing that we're going to take advantage of, right? That's the basis for gel electrophoresis, is that DNA is negatively charged. Now, let's say that we apply an electric current through this. What will happen is DNA will actually travel towards the positive electrode when exposed to an electrical current. So maybe you guys have jump-started a car before and you know that there's a plus and a minus, right? If we apply this current and we put DNA up towards the top, right? And we separate these currents and we say, like, well, let's run an electric current from negative to positive. If our DNA is negatively charged, it's going to repel away from this negative end, and it's going to be attracted towards this positive end, which means DNA will actually move or migrate when it's exposed to an electric current. We can push it, okay? We can push it in this direction from the negative, and it'll be attracted towards the positive if we apply an electric current, which we're going to. So it relies on some electricity as well. Small pieces of DNA, right? So let's say that you have 100 base pairs. Small segments of DNA will travel a lot further than larger pieces. So let's say 1,000 base pairs, right? Imagine that you had a tube and half the DNA has 1,000 base pairs and the other half is 100 base pairs. If we put it all in the same starting block and we say, okay, start, and we start the electric current, the 100 base pairs is going to travel way further, because in the same amount of time, that bigger strand of base pairs is going to go more slowly, because it's got more mass, it's bigger. It's going to have a harder time moving, right? It takes more force to generate something to travel further. So the small segment here is going to travel further, and the larger segment won't travel as far. So what that allows us to do is, if we have a mixture of DNA, it allows us to separate that mixture by size of the strands, which is awesome, and that's exactly what we want to be able to do. So let's take a look at some practice here. What does electrophoresis look like? Well, to do electrophoresis, the DNA must have something in which it can travel, right? It doesn't just travel through the air. So we've got to actually separate it inside of something. And what we're going to separate it in is a gel, which is how it gets its name, gel, electro for electricity, phoresis, to separate, right? So this gel is not unlike Jell-O. It looks the exact same. It's kind of floppy. It's clear. And it's made of a couple of things. Agarose, which is the Jell-O kind of like part that makes it kind of like a floppy solid. and then some salts, and that's going to provide a place for the electricity to travel through. If we have negative and positively charged things that salts have, electricity can travel through them easily. Okay, so then what we're going to do is we're going to place the gel inside of a gel box, which is going to have a special type of salt water in it, again, to allow current to travel through it. So that salt solution is going to be present in there as well, as you can see here. So here's the gel, the agarose gel, and then here's the gel box with the special salt water in it. Okay, DNA fragments then are going to be placed in a well at the top of the gel, and that's what you're seeing right here. This is the well, and that's where we're going to load up DNA. So what we actually do is we take your sample from something like PCR, and we put it in a micropipette, put it in a tip, and then we actually just put it right into that well right there. So all your DNA just sits there, right, and it's ready to run. When electricity is applied, that DNA is then going to travel through the gel. It's going to get pushed through by electricity through this gel. So we have to have a power source connected, and then we'll have the negative lead, or what we call the cathode here, and then the positive lead, or what we call the anode here. And we're going to run the gel, and then DNA is going to push its way through this jello sort of matrix. And remember, the smaller pieces will go further, the larger pieces will stay towards the top. Okay, so yesterday I came into the lab and got everything set up. So these are all of our samples from the PCRs that you guys ran, and this is a lot. So every single one of these samples has to have one of these little wells. So I took a micropipette, and you guys can see them back here even though they're kind of blurry. I took a micropipette, I got 10 microliters out of every single one of these wells, out of these tubes, and I loaded them into their own individual wells, right? And then you guys can see back here, these are what the chambers look like. Okay, so right here, maybe you guys can see this. There's like this little clear portion. And that little clear portion, that's the actual gel. And then I loaded up your samples in the wells up top here. And then you guys can see back here, there's a power supply that's actually going to supply the current. This is the negative end, and this is the positive end. Once I hook this up and I turn this power supply on, it's going to run through. And you can actually see that in a video that I'm going to show you here in a second. So what we hope to end up with is we need to visualize the DNA in a gel, but remember that that DNA is going to be invisible. Well, is there some sort of molecule that we've used before that's going to allow us to visualize DNA? And the answer is yes, right? It's invisible in solution, so we need to add ethidium bromide to it. And you guys remember you did the ethidium bromide spot test, right? Well, we're just going to do that again, but instead what I like to do is I just add the ethidium bromide directly to the gels while I'm making them. So I've actually included a video here of me a few years ago making a gel, what the process of making a gel looks like. Click on that so you get an idea of what making a gel actually looks like. You'll also do that in the simulation that you're going to do today. If we've got some ethidium bromide, what we can do is in these gels, right, like the DNA will get pushed through, and remember the bigger segments are going to stay up top, and then the smaller segments are going to travel further. And we get this separation that starts to happen inside of this gel. Now this gel is in a different type of technique. This wasn't done in a PCR, so there's multiple fragments, and we'll talk about why. Remember, today I just want you to understand how electrophoresis works, and then we'll talk about how do you read this data? How do you read these gels? What are they actually telling us? But what I wanted you to know is that we're going to use ethidium bromide again, and that's going to be sitting inside of the gel, so that when DNA gets pushed through it, we can see it if we apply UV light to it. Now, here's a video of your actual gels being run yesterday. So let me click this. And guys, notice when I hit the power supply what's going on here, right? So this is the negative end of the gel box. Positive end is here on the other side. And then when I start applying current through it, you can see all these bubbles start to appear. And that's actually hydrolysis happening. That's water splitting into its separate gases of hydrogen gas and oxygen gas. So what you're seeing is the current actually starting to run through, right? And it's going, and the power starting to push this DNA through. And then I usually run these for about an hour, depending on what I'm trying to find. So you have that much current pushing DNA through that gel for about an hour, they're going to separate, right? And that's kind of the goal of the whole thing. So let's see it, one more time here. Cool, awesome. And these are some of the actual results that we got yesterday. So primarily, I mean, these gels, like I said in the announcement, these gels look beautiful. These look awesome. So these are just 3 of the gels. I had to run a total of, oh man, 10. I had to run 10 gels yesterday, which was painstaking, to get these results. All right, so we'll talk about why we're seeing these results that we're seeing. But basically what you should know right now is you can actually see the little wells up here, right? If DNA got pushed through and it's moving through, you'll see it as this kind of banding pattern here. And the brighter spots that you see, the brighter bands, mean they have more DNA in them. The best gel out of these 3 would be this one. This one looks amazing. And tomorrow we'll talk about why. Why do they look so good? And why am I saying that they're looking good? And then how do you also read all of this data? Okay, so I hope that helps. Now you guys are going to go move on to a gel electrosimulation where you'll kind of get to practice it a little bit and answer a few questions. And then tomorrow we'll learn how to read these gels.